Anyone is waiting for joining? No. Okay. I guess we can start it. So this is another RNA virus family, Arteriviridae. So this Arteriviridae is a newly formed family of viruses. Especially if we say it's probably in 1980s only this particular family name has been given. Some of the organisms and some of the viruses included in this Arteriviridae family were recently identified. Of course, one or two members, the disease descriptions were there earlier. They have been put it in some other family, and recently in 80s they classified into a new family, Arteriviridae. So if you try to see it, then it is quite clear that some things are one of the classical disease under this family is the equine viral arteritis. Some sort of inflammation of the arteries takes place. So as such, the name has been given, the Arteriviridae. The other closely related viruses that include the porcine reproductive and respiratory syndrome virus, PRRS, as you know, that become in the limelight as in 2013. First time the disease entered through Mizoram to India, and it has causes a severe economic losses among the pig farmers in terms of mortality and reproductive problems. So in your syllabus, unfortunately, they mention only about the equine arteritis virus, which is not prevalent, I should say. In long time back, one serosurveillance was there, and few equines were found positive, seropositive only. After that, there is no record of equine arteritis virus in India. However, this the PRRS virus, which is occurring in our country from 2013 onwards almost every year. So I'll be focusing on both the disease and giving more emphasis to PRRS because we should know about this disease. So this Arteriviridae family, this is a screenshot. I've taken it from the ICTV. My intention is to make you aware about this ICTV web portal, and as and when if you feel some updating is needed, then you can go through it and for more clarification. Of course, this is so frequently they are changing the names and classification. Sometimes it is become confusing. So at any point, if any confusion arises, then you can refer here to for further details. So I'm just cropping it viruses of our importance under this family Arteriviridae. Again, these are similar to that of the coronaviruses that what we discussed. They are like close relatives of coronavirus Arteriviridae family members. They are under the order Nidovirales. So when we say Nidovirales, all of you will understand. It's a group of viruses. They produce nested set of messenger RNA, nested set of subgenomic messenger RNA, having a common 3' aim. So overlapping messenger RNAs are produced. So that's why they are called as Nidovirales. So similarly, here also the Arteriviridae family member, they also produces the nested set of messenger RNA during the virus replication. So out of 6 subfamily, 2 Equarteriviridae and Variarteriviridae are important for us. Equarteriviridae that include alpha Arterivirus genus that include the equine viral arteritis. It was earlier known as equine viral arteritis. Now the species name has been given as alpha Arterivirus equid. So as per the recent classification, that causes an equine viral arteritis disease exclusively occur in equine species, mainly the horse, donkeys, mule, you will see. And the second subfamily, the Variarteriviridae, That include the beta arterivirus, and beta arterivirus and the subgenus Empoba arterivirus include PERS2 virus, which is recently reclassified as beta arterivirus suite 2, which is prevalent in our area in India and Southeast Asia. We have this beta arterivirus suite 2 infection, the PERS2 virus. Whereas in most of the European countries, they have the Europa arterivirus subgenus, beta arterivirus suite 1, PERS1. So PERS1 and PERS2, these are the two members of porcine reproductive and respiratory syndrome virus, which nowadays call as beta arterivirus suite 1 and suite 2. So out of these two, the suite 2 is the typical American type, and this is prevalent in our country as well as in Southeast Asia. And the beta arterivirus suite 1, it is more prevalent in European countries. They have this virus infection. Okay, so this is about this classification. So we'll go to the general characteristics of this family. Arteriviridae family general characteristics. So we'll find they are the envelope viruses with helical nucleocapsid. Sorry, the icosahedral symmetry. Remember, this is not helical. Icosahedral symmetry, and their envelope viruses, and the size is meter in diameter, not much bigger, and the genome size varies, like 12.7 to 15.7 kb. So in comparison to the coronaviruses, their genome size is quite smaller, and they carry the genetic information for the important proteins and glycoproteins, like 10 open reading frames are there, and they give rise to different structural and non-structural proteins. So some of the structural proteins important for us is the glycoprotein Z. And this is highly immunogenic, against which the vaccines are targeted, as well as this also provides it helps the virus to land on the cell surface for initiating infection. Nucleoprotein N and matrix protein, they are associated with the viral nucleic acid. So they are positive sense single-stranded RNA virus, non-segmented. Okay, positive sense single-stranded RNA virus. So this positive sense single-stranded RNA virus, the host cells, they can initiate the protein synthesis, but at the genomic replication stage, they produces the nested set of messenger RNA having common 3' end. Okay, so if you try to understand this particular pictures, then you'll find this is the entire genome length of 12.7 kilobases from 5' to 3' end. 3' is contained in this poly A tail. And these are the different reading frames, and this depicted the individual genes and the proteins are produced. Now, if you see it minutely, then you'll find this area, which is depicting most of the structural protein. The messenger RNAs are overlapping. They are having common 3' end. These areas are common in all the messenger RNA. Means this particular portion depict all the important structural proteins, and the messenger RNA are overlapping in nature. That's why they are called as messenger RNA, having common 3' end. Okay, so same genomic sequences, but coded for many different proteins. This type of characters we could see among this certain viruses. So in eukaryotic system, this kind of nested messenger RNA are never formed. So that is why the corona and arteriviruses are grouped under the nidovirales order. So why they are called as nidovirales? Because they produces a nested set of messenger RNA, just like the bird nest, crisscrossing messenger RNA are produced, having a common 3' end. These are all overlapping. That means this particular sequence and this one is totally within this sequence, but the proteins are different. So in different reading frame, the protein translation takes place. Okay, So if you talk about the nature of the virus, then you'll find the virus, they are really cannot survive for long. So usually associated with the infected animal body, or maybe in the carrier animal, the virus remain inside the body, and once it comes out, they readily get inactivated. Since they are enteroviruses, they are sensitive to the lipid solvents. This virus, they purely grows in the cytoplasm of infected host cell. There is no any nuclear sites for replication. So the virus under this group, we can grow in the cell culture, primary cell culture. However, they show some specificity. They are not readily adapted to all type of cells. More particularly, if you talk about the PRRS virus, PRRS virus, they are highly specific, like one receptor is called a CD163 molecule is essentially needed for initiating the infection. That is why they have limited tropisms, and we have to go for a specific primary cell line called as pores and alveolar macrophage cell culture, where we can grow this PRRS2 virus. However, some cell lines like MARC145, these are modified cell lines which express this receptor site for the virus, can be used for propagation of the virus. So do not produce cytotoxic effect. Of course, it is not a generalized statement. Some strain, of course, they can cause total disruption of the cells. However, some members do not produce, particularly the PRRS1 virus doesn't cause us any cytotoxic effect. Now, coming to those are the general characteristics of the family Arteriviridae. Coming to the respective disease, as I said, this disease has not been mentioned in your syllabi, unfortunately, but we must know about this disease, as because in 2013, first time the virus infections we could record in the state of Mizoram, and thereafter it spread to different districts of Mizoram. We heard about some outbreaks in state of Meghalaya also, but later on they have not recorded any further outbreaks. But from 2013 onward, consecutively, almost every year, we are recording outbreaks of PRRS in Mizoram. The city at which the earlier outbreak occurred gradually is declining, and number of outbreaks are lesser than that of the previous time. So it's a good thing that the virus has not been transmitted to the other part of the country. So this disease has already been recorded in European countries and American countries. As I said, this is comparatively a new virus for us. Probably somewhere in 1986, the disease has been recorded, and immediately they could identify two members. One is the PRRS1 and PRRS2. Very interestingly, although they are a virus, but there are a lot of genetic differences to be seen among these two viruses. So PRRS1 and PRRS2 virus, they are hardly genetically 60% related. Usually it doesn't happen with other viruses, and they cannot cross-protect each other. So since its prevalence of beta PRRS1, some vaccines are available in the international market, but the PRRS1 vaccines cannot give protection against PRRS2, and vice versa. This is the story. And because of the nature of the virus, it is difficult to get some live attenuated strain, and vaccine become a constraint for us, and it's not available in Indian market as of today. However, in international market, people they use some vaccines to control this infection. So this disease, some the synonyms are peers, porcine epidemic abortion, and respiratory syndrome. As because this virus, the main clinical manifestation is severe respiratory distresses, and in pregnant animal abortion. Abortion is inevitable, always it will occur. The initial respiratory tract infections will give rise to severe disease severity, and then during the circulation, the viruses causes endothelial damage, and that ear pig disease. In white skin pig, we can see the ears are cyanotic, appearing as blue color. That's why sometimes called as the blue-eared pig disease. So this is basically a cause of the arteritis condition that occur in the blood vessels. Okay, so if you try to analyze the clinical manifestation and describe the disease, then we must mention that this is a systemic viral infections that predominantly exhibiting reproductive failure in breeding sows and gills, then increased pre-weaning mortality and respiratory problems. So they are the organism PERS1 and PERS2, and please remember, in our country we recorded only the PERS2 virus. For many years, the disease descriptions from USA and European continent like Germany, Netherlands, and many other European countries like Austria, Belgium, they encountered this disease. And most of the time it was found to be disease restricting to the respiratory and reproductive problems. The mortality was not recorded high at the time, but in 2016, a particular variant appears in again Southeast Asia, particularly in the China, and that become highly virulent. And that particular strain has been given name as HPPRS. Means highly pathogenic porcine reproductive and respiratory. PRRS virus can cause large-scale mortality that we encountered in Mizoram. Along with the reproductive and respiratory syndromes, there are large-scale deaths observed in PERS disease because of the HPPRSV. So likewise, since it is an RNA virus, it born every times when it propagates, and it changes its tract of the production of the disease. Most of the time, the viruses try to escape the immunity and try to produce the disease in a different form. Likewise, the variants and mutants are produced frequently. So this disease, you'll find it's exclusively occur in pig. No other species the disease has been recorded. And in the pig, since it is causing the respiratory distresses and abortions, so the disease transmission is mainly occurred through airborne transmissions or aerosol transmissions. So close contact through droplets, the disease transmission may occur. Even it has been seen the breeding board, if get infected, the virus can be disseminated in semen, and using those semen for artificial insemination is also a source of transmission. Besides this, the in utero transmission also occur to the fetuses, and they behave differently immunologically to the PERS infections. So the virus, they are excreted in all excretion and secretions by the infected animal, right? So as I said, the propagations, the PERS virus, they are having a special cells, as called as the porcine alveolar macrophages. These cells, they express, originally they express the CD163 molecule, which has become the target site for PERS2 virus to initiate infection. So later on, that CD163 has been transfected to certain cells like MARC145, and these cell lines are also available for growth of this virus. Okay, so coming to the clinical signs and symptoms, the reproductive failure in pregnant sows and respiratory disease in young pigs, classically what we explain it. However, the HPPRSV causes additional death, mortality, high mortality because of severe lung damage that occur, right? So definitely that is also a kind of cytokine storming that occur because of the virus growth in those alveolar lining epithelial cells and the blood vessels, and they cause a severe inflammatory reactions in the lung. Okay, so in some cases, of course, Streptococcus suis, Haemophilus parasuis, Echinopsis pleuronimonii, this may aggravate the conditions and the mortality may increase, but HPPRSV, of course, they are not dependent on this organism. As such, it can cause large scale death, right? So some of the clinical manifestations will be some patchy hemorrhages we can see in the body surfaces, and the rapid rate of respirations, lateral recumbency, and high fever, and finally death occur, right? So in pregnant animal, obviously, because of the arteritis conditions, they will abort. So stillborn, mummified, and decomposing piglet is an that we can see it affected heart. Then the growing pigs, particularly, who are mostly affected, the necrotizing pneumonia and consolidation of the lung takes place. So when these inflammatory reactions that takes place in the lung, the inflammatory exudates are accumulated. So because of which there is consolidation of the lung. The main function is almost similar to that of the pathology, what we understand about this SARS-CoV-19 infection. SARS-2 virus infection, there also this consolidation of the lung takes place. As a result, the animal, they suffer from hypoxic conditions, low oxygen tension, and they die, right? So in certain cases, strains, we can see the accumulation of fluid in the abdominal cavity, even in the thoracic cavity. And this is because of this arteritis condition. The blood fluid leaks from the vessels, not the cells, only the fluid part. As a result, we can see the stroke-color fluid accumulation in the abdominal cavity. So these are some of the pictures we can see why they are called as the blue ear, the mummifications of fetuses that occur because of in utero death of the piglets and abortion, stillbirth. These are the frequent outcomes of PRRS. Looking at it, we can get an idea about the prevalence of PRRS disease. So it affects all the pigs only, accidentally infected animal, and they become a source of infections, particularly the boars. They may save the virus in semen for a period of time, and they may again causes a new fresh outbreak in the area. So when you talk about the diagnosis of the pathology is all like depend on that the arteritis conditions, the epithelial cells inflammations lead to those cyanosis and abortion condition. So clinical signs and symptoms is one clue, but it is not conclusive because there are many viral infections where we can see similar kind of patchy hemorrhages and bluish, cyanotic bluish ear, or abortions in pig, like classical swine fever is another example. Even the swine influenza also, same kind of things may produce. Even certain bacterial infections like Pasteurellosis, sometimes it is confusing, Bacillus pleuroniae, mycoplasma. So clinical science is not very much conclusive. We have to go for laboratory diagnosis. In the laboratory diagnosis, the most easiest and quickest test is the nucleic acid-based detection. So we go for the RT-PCR test to detect the presence of the virus clinical samples. And this is the regular process what we are doing in our laboratory. Besides this, we also isolated the virus in porcine alveolar macrophage primary cultures, and we can propagate them and confirm them by fluorescent antibody test and ELISA in our laboratory for virus detection. So at the same time, the serodetection is another aspect. Of course, the area where the disease is endemic, the sero detection doesn't play an important role in disease diagnosis, because some of the animals may show seropositive, maybe because of previous exposure in previous outbreak. So on the basis of serodetections, we cannot give a conclusive diagnosis. So all the times our main focus is on the virus detections, either by different immunological tests like FAT, ELISA, immunoperoxidase test, or in situ hybridization test, or we can go for the nucleic acid based detection along with the isolation process. So this way we can diagnose the disease with confirmation. So this is one slide showing this porcine alveolar macrophages infected with the PRRS virus, and the cells are producing apple green fluorescence. They have used the FITC conjugate for staining. So if the reagents are readily available, so instantly we can detect it, and within say 1 to 2 hours we can get the confirmatory results. Now, if you talk about the prevention and control, unfortunately, as I said, that we don't have any vaccines as on today's date in our market, in Indian market. However, in international market, the vaccines are there, where people mostly use the killed oil adjuvanted vaccines, because this is a safer vaccines. No doubt, it cannot give very good protection. Live attenuated vaccines are marketed in European continent, but live attenuated vaccines, which will not be effective in our case. For instance, there are 60% dissimilarities there. So one cannot literature, then you'll find some killed oil adjuvanted vaccines using the PRRS2 virus are also available in Southeast Asian countries. However, the vaccine has not been country as on today's date. So one more important point is that none of these vaccines can confirm the total virus clearance from a flock. If you give the vaccines, you cannot say that the virus will not persist in the flock. So it can reduce the disease intensity, but cannot make sure that the total clearance or sterile immunity, what we call, cannot be achieved by this kind of vaccine. So the countries, they usually go for the vaccinations and other hygienic measures and strict biosecurity to maintain it so as to avoid this disease outbreak. So some of the things that what we should do along with those biosecurity measures, particularly in the breeding stock, if we introduce the animal, we have to be thorough and test it very properly with antibody test, whether the animal has been exposed previously or not. Only then it should be introduced to the stock. Otherwise, they become the source of infection most of the time, what we could see. Then the strict biosecurity measures that we can follow it. Once the outbreak occurs in certain locality, immediately we have to cordon those area, and we have to restrict the movement of human from farm to farm, because that process the disease transmissions may occur. No doubt the virus is not very much stable in the environment, cannot survive more than 1 day outside the animal body, but through human carrier, it can be transmitted from one farm to the other farm. So these are some of the vaccines. I just took the clips from Google, and these are available in the international market, not available in our country. Now coming to the second infection, equine viral arteritis, and because of this disease, the whole family name has been given as Arteriviridae family. Recorded long time back in 1953 in USA, but the virus could not be classified in some family because of certain regions, and finally in 1980s only when the Arteriviridae family was developed, and this member has been kept in that particular group. So this virus, they basically causes conjunctivitis and edematous conditions in the body because of the arteritis conditions, and in folds, basically it causes severe respiratory infection. So the death is not very high injury. This cases may aggravate with other bacterial infections, and that may lead to some mortality may appear. So commonly it is called as the pink eye disease. What I say that you can have a notebook and write the name of the disease and some synonyms like pink eye disease and the quality of organism, these things you can record it. As I say that no doubt the disease has been recorded from many countries, but in India, many years back, some seropositive animals were detected in army start farm in Meerut, both in UK as well as both in UP as well as in Haryana. They detected few seropositive cases, and interestingly, those 15 seropositive mares is having a history of abortion. So after that, no such incidents has been recorded in our country. Considered to be an important viral disease of equine. So the Alpha Arterivirus equid is the etiology, and Alpha Arterivirus genus, subfamily Arteriviridae, and family Arteriviridae. So the equidae means the horse, donkeys, and mules. The disease has been recorded, and it is basically transmitted through both respiratory and reproductive tract, like aerosol and direct contact. Aborted fetus and placenta excreted large amount of viruses, and other in contact animals directly pick up the virus from the environment. Even the venereal transmissions may occur, sexually transmissions may occur in this particular instance. So basically through 14 days of incubation period, we could see the rise of temperature, and then leukopenia obviously caused leukocyte infections, and then the prominent is the edema of the hind limbs, then eyelids, then conjunctivitis, and if it is a pregnant animal, the abortions occur. And the entire pathology is can be described as a the vasculitis occur that leads to exudation of fluid from the blood vessels, leading to edema formation. So sometimes in male animal, the scrotal edema becomes so prominent from distance itself, we can get some idea about the disease persistent. Secondly, this is another major issue. The persistent infections are seen in equine, and the virus, after the episode of the infections, the immune system of the animal will not allow the virus to produce the active infection, so they go for persistent infection. But this is a persistent productive infection. So at slow rate, the virus production takes place and are excreted through their secretions. So those animal, persistently infected animal, become a source of infections, and they may attribute to the fresh outbreaks occur in equine species. So some edematous swellings and abortions. So here, these viruses can be propagated, doesn't have much specificity for certain cell line, unlike the PRRS virus, what we discussed. So they can be grown in rabbit kidney 13, Vero cell line, BHK21, even the equine fetal lung cell culture also it can be propagated. But they need around 2 to 8 blind passages to start developing the therapeutic effect. So initially they do not produce any changes. Gradually, when the virus get adapted, they start producing some of the rounding and shrinkage of cells and total disruptions. Likewise, this the three sequences of cells showing here. This is a normal monolayer of RK13 cells, and here the cells is producing the rounding and disruptions starts, and finally the whole monolayer disrupts off from the flux. So this way the virus growth, virus can be grown in the cell culture easily. And in the diagnosis process, we can go for all these methods every time we discuss it, including the molecular method for diagnosis. No specific test recommended. All are equally good for confirmatory detections. Differential diagnosis will be needed because we are having certain equine herpesvirus infections where also rhinopneumonitis and abortion occur, similar type of clinical manifestation. Even we talk about the equine African horse sickness. There also you could see the respiratory distresses and edematous swelling. So you have to go for differential diagnosis. By molecular method, no doubt, with accuracy we can take the growth of the presence of the virus. So since the disease incidence itself is not there in our country, obviously no vaccines are available in our country for vaccinating the animal, commercially available. Of course, some private stud farms, it's growing up in our country. The breeding farms, they may use it, but there is no record we could see of using this vaccine certificate. So both modified live and the formalin inactivated vaccines are preferred for, both vaccines are available for vaccinating against this equine viral arteritis. So these are the two infections under the Arteriviridae family. So I will request you to go through the PRRS infections from Lux, because this is a really important disease for us. Well, tomorrow, the afternoon half, combined class, Suri sir will take it on the different detection method for virus. As we are discussing about this, all different tests like immunoperoxidase test, AZPT. So he'll give you a brief idea about the whole diagnostic test that we can apply for viral disease diagnosis. Okay. So after that, again, I will continue on Friday.